Welcome back to Getting Started in CellEngine. Today we'll be discussing some of the advanced gating tools available to make gating faster and easier. If you haven't watched the earlier videos on setting up your experiment and basic gating in CellEngine, you might want to watch those first. They'll be linked in the video description below. There are several important features we'll discuss today, including importing information from another experiment in CellEngine, making Boolean and combination gates, and gate tailoring, which is the process of adjusting gates sample by sample. Tailoring can be done manually or using CellEngine's built-in machine learning algorithm, and we'll go through both of these options as well as how to gate by percentile. So let's go to CellEngine, where I've got a new experiment open. I've already uploaded some FCS files and added annotations, so we're ready to do some gating. However, this time we're going to import some information from an experiment similar to this that I've already analyzed. To do that, I'm going to go down to Tools on the left-hand side of the gating page and select Import. This dialog will ask you to select a source experiment to import from. Mine is in my reference folder, so I'll click to see the contents and select the TH subset experiment. Once you've selected a source experiment, you'll have the option to import populations. I want to import populations to save time on gating, so I'll check the checkbox here at the top of the page. CellEngine will match any channels that are the same between your two experiments, but you can reassign those as necessary so you don't have to have the same panel for both. For example, if I had CD3 on red 670 in my source experiment, but I changed the fluorophore for this run, I could click on the drop-down and reassign it. When the channels are properly matched, click on Next. If you're importing populations, you can also import illustrations. We haven't talked about illustrations yet in this series. That will be where we'll create figures after gating is finished. We'll go through those options more in depth in a future video, so for now, I'm going to skip this option. Scales and compensation matrices can also be imported. You can import these regardless of whether you import populations or illustrations. I'm going to go ahead and include both of these. Once you've got the options you want, click Import. My previous analysis was set up to look at this T helper cell population. I'd like to look at the same phenotypes in the Tregs. To do that, I'm going to right-click on the TH gate. Right-clicking gives you a variety of options for the gate that you have selected. In this case, I'm going to copy the gate, which will also copy all of its children. Then I'll right-click on the Treg gate. There are three basic options for pasting gates available. You can paste the gate that you copied along with its children. You can paste the gate without its children, or you can paste just the children of the copied gate. In this case, I want the subpopulations only, so I'll choose the last option. Once you've decided which gates to paste, you can choose between pasting them linked or unlinked. Unlinked gates, just like gates you'd create manually, will move independently from each other. Linked gates will share the same position, even in different hierarchies. I'm going to select linked for this example. Now, if I move the CD28 gate for the Treg population, the CD28 gate will also move for the TH population. When you have a gate selected, any gates it's linked to will be bolded in the hierarchy, so they're easy to see. Right-clicking gates will also give you other useful options. If you right-click on a rectangle gate, you can convert it to a polygon gate. If it's a polygon gate, you'll be able to add an additional vertex that you can place. If you end up with too many vertices, remember that you can select them individually and hit delete to delete them. Boolean and combination gates are also created from this right-click menu. You can select multiple gates by shift-clicking. You can also use the Control or Command key if you're using Windows or Mac respectively to select multiple gates. Boolean gates contain and, or not combinations of the selected gates. Not gates can also be created just by selecting one gate. Unlike Boolean gates, which just create one population, combination gates create every permutation of the gates that you have selected. This can be especially useful if you're doing something like multifunctional cytokine analysis. Up to 12 gates can be used for combination gating at a time. One word of caution, the number of gates created is 2 to the n, where n is the number of gates that you had selected originally. So this can add large numbers of gates to your experiment. You can always save a revision of your experiment before adding large numbers of combination gates. You may remember from a previous video that a revision is an immutable snapshot of your experiment, and it can be created at any time from the summary page. Now that we've added the gates that we want to the experiment, we may want to make adjustments. By default, a gate in CellEngine is the same shape and position for every sample. However, sometimes sample variability means that you need to adjust the positions of gates independently. You may encounter this more in CellEngine because it can handle large numbers of samples overall, so you may find it useful to have different cell types or more disease states in a single experiment. Tailoring allows for adjustment of gates independently for each sample in an experiment. In this case, it looks like the position of the gate varies between these samples. To turn on tailoring, select the gate and then click on Enable Profile Tailoring on the left. You'll notice that a superscript T appears next to the gate name in the hierarchy. That means tailoring is enabled for that gate. Now I can change the gate on one sample and not affect the others. You can adjust each sample individually one by one, but often you'll want to use the same position for multiple files in an experiment. There are two ways to approach this. You can copy a gate by right-clicking and then right-click again to paste the gate geometry onto another sample. However, you will often want to apply the same gate position to larger groups of files. For example, if donor variability is the reason for differences in your gate positions, you might want the same position for every sample from the same donor. To do this easily, click on Apply Tailoring To on the left. From here, you can select specific files from the file list, filtered by annotation information. You can also select any files that share the same metadata. This is another reason that adding sample annotations early in the analysis process can be helpful. Because I added donor IDs before gating, I can use them to select other samples. When you have the settings you want, click on Apply. If you ever want to check and see which gates are in the same position for different samples, there's a tool on the left called Analyze Gate Tailoring. This will open up a window that will show all of the tailored gates as columns and all of the FCS files as rows. Each file will have a value for that particular gate that will be either G, indicating that it's in the global position and hasn't moved since tailoring was enabled, or an integer. Any two files with the same integer have the same geometry for that particular gate. This can be helpful for double checking to see if tailoring was applied properly to all of your samples. When you have large or highly variable experiments, manual tailoring can be extremely time consuming. CellEngine has an add-on tool that will automatically adjust gate positions. Before going into an example, let's look at an overview of the process. CellEngine's auto-tailoring tool uses a machine learning algorithm to adjust gate positions. You provide the algorithm with one or more manually tailored files to train it on gate placement. The algorithm will attempt to make similar choices in gate placement as the reference files. So you'll initially select a set of files that is representative of the rest of the experiment and then manually tailor them. I usually begin with one sample from each experimental condition. For example, if my experiment includes several cell lines, I might include one file from each cell type. Then you'll select the files you want to use as reference and the files you want tailored, and the auto-gating algorithm will adjust gates for you. This creates gate positions that are functionally identical to those that a human analyst would make, so you can review and adjust them just like you could your own gates. Let's look at an example in CellEngine. I've got an experiment open with a lot of variability. Here are some dot plots of various files in the experiment. Gating by hand would require adjustments on essentially every file. I've selected several representative files from my conditions, A and B. You don't actually have to mark these in any way, but adding a column in annotations to indicate if you've chosen a file as part of your training data will make it easier to select and provide a useful reference down the line if you need to come back to an experiment in the future. In gating, I've gone through my training set and manually adjusted each gate to be where I want. You can auto-gate multiple gates at the same time, so when I'm ready to proceed, I'm going to select the two gates that I want to tailor and click on Auto Tailor. Here, I'm going to choose two subsets of files. First, I'll pick the reference files that I manually tailored. Since I added a column in annotations, I can just filter the list based on the training value. And when I've selected the files that I want, click the box at the top of the column to select all filtered files. Then I'll do the same for the target FCS files. If you want files to share gate positions, you can choose to tailor by one or more metadata categories. For example, you might want to tailor by donor or by cell type. In this example, let's tailor each file individually. There are three algorithmic variables that you can adjust. Minimum improvement is the minimum amount of improvement that would be needed in order to adjust a gate position. Minimum events is the smallest number of events in a population that needs to be present for the algorithm to run. Smoothing variance affects how much smoothing is applied to the results, which can reduce artifacts and improve result outcomes. The default settings work well with almost all biological data and are a really good starting point. When you're done with setup, click on Tailor. Auto-tailoring is a rapid process, usually completing in a few seconds for gates from the same part of the hierarchy. In addition, the algorithm doesn't randomize any part of the results, so if you give it the same training data on two different runs, you'll still get exactly the same results. These features make it easy to work iteratively and add new files to the dataset if you need to. To review our results, let's look at the dot plots with gate positions. We'll talk more about how to set these up when we discuss illustrations. As I scroll through samples, if I see gates that I'd like to adjust, I can right-click on the plot to go directly to that file in gating. Because auto-tailoring is so quick and because there are other advantages to iteration, it lends itself very well to this process where you can adjust a gate and then add that file to your training data and rerun the algorithm to improve your outcomes. As I mentioned, automatic gate tailoring is an add-on feature to CellEngine. If you think it would be useful in your research and you'd like to try it out or add it to your subscription, let us know. The last topic I'd like to touch on in this video is percentile gating. In some cases, it's helpful to be able to adjust positions based on percentiles. For example, you might want to look at the top quartile of cells producing the most of a cytokine, or set a gate to include any events that are over the 99th percentile of a control. Each vertex in a gate can be moved to a percentile from the same file, another file, or an average of a group of files. As an example, let's look at our earlier T-cell experiment. We'll adjust the PD-1 gate based on percentile values. Any vertex in a rectangular, polygon, or quadrant gate can be adjusted in this way. First, make sure you have tailoring enabled for the gate, then right-click on a vertex and select Move to Percentile. This will open a new dialog. We'll select the files that we want to tailor the gate on. For this example, let's tailor the experimental and control file from these two donors. Those happen to be the top files in the list, but we could also use annotations to filter files for selection, just as we did earlier. You can choose whether to tailor by one or more annotations. For this example, let's tailor by donor so every file from the same donor gets the same placement. Next, we'll decide how to select control files. First, pick the appropriate annotation column, in this case, time point, then select one or more values. For this experiment, SCR is the proper control. Then we'll choose our percentile value. I'd like to set the gate to include the top 20% from the control samples, so I'll set the value to 0.8 and click on Tailor Gate. There's only one control sample for this experiment for each donor, so the gate on the control will always be set at the percentile threshold. If there were multiple controls, an average of the control data would be used to set the position of the vertex. The experimental sample from the same donor then has been set in the same place, so now we can compare each donor back to the control. In some cases, the distribution of data will make it impossible to set the gate at exactly the percentile value. In those cases, CellEngine will set the value as close as possible. I hope this overview of advanced gating tools has been helpful. In the next video in the CellEngine series, we'll move on to illustrations and make figures with the data that we've analyzed. If you have any questions in the meantime, please reach out to us at support@cellengine.com.